Good morning, everyone, and welcome. I'm excited to have you here today. We'll be exploring the topic of statistical optimization of in-process sampling during sterile manufacturing. My name is Dr. S.K. Singh, and I hold a PhD in this field. In this presentation, I'll guide you through how we can improve sampling processes using statistical methods, ensuring better quality and efficiency. So let's dive into this important subject together. Welcome to this section on sterile drug products. When we talk about these products, it's crucial to understand that they require very strict in-process controls. These controls are essential to ensure several key aspects. First, sterility itself, because any contamination can compromise patient safety. Second, product uniformity, so every dose meets quality standards. Third, the absence of particulate contamination, which can affect both safety and efficacy. And finally, container closure integrity, to prevent any external contaminants from entering the product. At the same time, we need to be mindful of the sampling process. While sampling is necessary for quality checks, excessive sampling can lead to some issues. It can cause product wastage, which is costly and inefficient. It also increases the risk of contamination, as each sample drawn is a potential entry point for contaminants. And of course, more sampling can elevate operational costs due to additional resources and testing. This is where optimized sampling strategies come into play. They help us strike a balance, maintaining the necessary sterility assurance while minimizing the amount of product we need to sample. By doing so, we reduce waste and contamination risk, all while keeping costs in check. Ultimately, effective sampling strategies are a key part of ensuring product quality without compromising safety or efficiency. Let's talk about the regulatory requirements that are essential for sterile manufacturing. First, we have FDA 21 CFR 211.11, which emphasizes the importance of having written procedures for in-process sampling. This means you need clear, documented methods to collect samples during the manufacturing process to ensure quality at every stage. Additionally, these samples should be representative of the entire batch, giving us an accurate picture of the product's consistency. Next, there's FDA 21 CFR 211.16, which highlights the need for scientifically sound sampling plans. This is about designing sampling strategies based on solid scientific principles, ensuring that our sampling methods are both effective and reliable. All of these requirements come together to support the core goal of sterile product manufacturing, preventing microbiological contamination. To achieve this, sterile products must be produced using validated aseptic processes. Validation is critical because it proves that our processes consistently maintain the sterile environment, safeguarding patient safety. So, in essence, these regulatory requirements guide us in establishing robust procedures and scientifically supported sampling plans, all aimed at maintaining the highest standards of sterility and quality in our products. Let's take a closer look at the concept of risk-based sampling. Essentially, this approach focuses on identifying and monitoring the critical process parameters, or CPPs, that have a direct impact on the critical quality attributes, or CQAs, of our product. By targeting these key points, we can ensure higher quality and consistency in our processes. To do this effectively, we rely on several important tools. One of them is failure mode effects analysis, or FMEA, which helps us systematically evaluate potential failure modes and their impact. Then, we perform risk ranking to prioritize which parameters need the most attention based on their potential risk. Lastly, process capability analysis allows us to assess how well our process performs relative to the target specifications, giving us confidence in our control strategies. The overall goal here is to focus our sampling efforts where they matter most, on those parameters that truly influence product quality, so we can enhance our process understanding and ensure consistent quality throughout production. This approach aligns with our broader theme of risk management and continuous improvement in our manufacturing processes. Let's take a closer look at the statistical optimization approach. This method leverages various statistical tools to enhance sampling and quality control processes. For instance, process capability analysis, which includes CP and CPK, helps us understand how well a process meets specifications. Control charts allow us to monitor process stability over time, quickly identifying any deviations. ANSI and ISO acceptance sampling standards provide structured guidelines for evaluating batches efficiently. And confidence interval sampling gives us a statistically sound way to estimate parameters with a known level of certainty. The key benefits of using this approach are quite significant. First, it reduces the number of samples we need to test, saving time and resources. Second, it ensures that our batch representation is reliable, giving us confidence in the results. And finally, it provides a solid scientific justification for regulators, which is crucial in demonstrating the robustness of our quality assurance process. Overall, this approach aligns perfectly with our goal of making data-driven, efficient, and compliant quality decisions. Let's walk through the typical sterile injectable manufacturing process. First, we start with raw material dispensing, where the necessary ingredients are carefully measured and prepared. Next is solution preparation, which involves mixing these raw materials to form the drug solution. Then, we move on to filtration, which removes any unwanted particles or contaminants. After filtration, sterile filtration ensures that the solution is free from microbes, maintaining the sterility required for injectables. Following this, we proceed to aseptic filling, where the sterile solution is transferred into vials or syringes in a highly controlled environment. Once filled, the containers are stoppered and sealed securely to prevent contamination. The final step is visual inspection, where each container is carefully checked for defects or particulates. It's important to note that each of these stages requires in-process testing. This ensures that every step meets strict quality and safety standards, which is essential for protecting patient health and maintaining product integrity. Overall, this process highlights the meticulous attention to detail necessary in manufacturing safe and effective injectable medicines. Now, let's take a closer look at the manufacturing process for eye drops. Typically, there are several key stages involved. First, we start with raw material dispensing, where the ingredients are carefully measured and prepared. Next, we move on to solution preparation, mixing the components to create the correct formulation. Once the solution is ready, it goes through sterile filtration to remove any unwanted microorganisms or particles, ensuring the product's safety and purity. After filtration, the solution is filled into bottles, which is a critical step to maintain aseptic conditions. Following filling, the bottles are stoppered securely to prevent contamination. Finally, each bottle undergoes visual inspection to verify quality and consistency. It's important to remember that ophthalmic products require strict microbial control throughout the entire process. Since these drops come into direct contact with the eye, any contamination could lead to serious infections or complications. That's why maintaining a sterile environment and rigorous quality control is essential in manufacturing these products, aligning with the overall goal of ensuring patient safety and product efficacy. Now, let's take a look at a typical example involving sampling and wastage for injectables. Imagine we have a batch of 100,000 vials. When producing such a large batch, it's common to set aside some vials for sampling to ensure quality and safety. However, sampling inevitably results in some wastage because those vials can't be used for the final product. Additionally, there's always some wastage during manufacturing, like losses during filling, capping, or inspection. These factors are essential to consider because they impact the overall yield and efficiency of the production process. Understanding the typical levels of sampling and wastage helps us plan better and ensure that we maintain quality while minimizing losses. It also highlights the importance of accurate forecasting and process control to keep wastage within acceptable limits. Let's talk about our optimized sampling strategy. As you can see, we're relying on validated processes to ensure accuracy and reliability. This means we've carefully tested and confirmed our methods work as intended. Additionally, we're using statistical justification to back up our sampling approach. This helps us determine the right sample sizes and ensures our results are both precise and representative. By combining these validated processes with solid statistical reasoning, we can confidently say that our sampling strategy is both efficient and trustworthy. This is a key part of our overall approach to delivering high-quality, reliable data. Today, let's look at some key data points. We worked with a total of 300 vials, which gives us a solid base for our sampling process. Now, when we talk about reduction, we're seeing a 40% decrease in sampling wastage, which is significant. This is especially important when we consider injectable batch optimization, and I'll walk you through an example. Imagine a batch size of 100,000 vials. Traditionally, to ensure quality, you'd sample about 50 vials every hour over a 10-hour production run, totaling 500 vials sampled. That's quite a lot, and it adds to wastage and time. But with our optimized sampling approach, we only need to sample 30 vials every hour. This means fewer vials are taken for sampling, and as a result, we reduce wastage while still maintaining quality assurance. This example highlights how smarter sampling strategies can lead to significant improvements in efficiency. It's an important part of our overall goal to optimize processes, reduce waste, and ensure high-quality production. Let's talk about the reduction achieved here. We've seen a 44% decrease in sampling wastage, which is a significant improvement. This means we're using our resources more efficiently and reducing unnecessary waste. Now, when we consider batch size, we're working with 5,000 liters of solution, which is quite substantial. Managing such large batches requires precision and control to maintain quality and consistency. As an example, let's look at eye drop manufacturing. It's a great illustration because even with small, sensitive products like eye drops, these reductions in wastage and optimized batch sizes can make a big difference in cost and quality. Overall, these improvements emphasize how streamlining processes directly impacts both efficiency and product quality, which is crucial in our industry. Let's take a look at some effective process monitoring strategies that help us minimize sampling frequency while maintaining quality. One key approach is process analytical technology, or PAT, which allows us to analyze processes in real time, reducing the need for frequent sampling. Next, we have statistical process control, or SPC, which uses data analysis to detect variations early and ensure consistent output. Automated particle detection systems are also valuable. They enable continuous monitoring of particles, helping us catch issues promptly. Continuous process verification is another important strategy, as it involves ongoing validation of the process to ensure everything stays within control limits. Lastly, environmental monitoring plays a crucial role in maintaining the integrity of the process environment, preventing contamination, and ensuring product quality. It's important to remember that when we perform sampling, we need validated procedures that are designed to avoid any contamination risks. This way, we keep our process safe, reliable, and compliant. These strategies, when integrated effectively, help us achieve a robust process with minimal disruption and optimal control, aligning perfectly with our overall goal of quality assurance. Before we move forward with sampling reduction, it's crucial to have the proper documentation in place. First, we need a thorough risk assessment report that evaluates potential impacts on product quality and patient safety. Next, process validation data is essential to demonstrate that our manufacturing process consistently produces quality products. We also need a well-defined sampling SOP, which outlines the procedures for sampling activities. Additionally, a solid statistical justification is necessary to support our sampling changes, ensuring that reductions won't compromise product quality. Change control approval is also critical. It provides documented authorization from the relevant quality and regulatory teams. Finally, we must keep detailed batch manufacturing records that trace each step of production. All of these documents together serve as the backbone for traceability and compliance with GMP standards. Proper documentation not only supports regulatory requirements, but also helps us maintain confidence in our manufacturing process and product quality. Remember, thorough documentation is key to ensuring that any changes we make are transparent, justified, and compliant. Let's take a closer look at this overall wastage reduction example. What we're seeing here is a practical illustration of how targeted efforts can lead to significant improvements. By analyzing each stage of the process, we can identify where waste occurs and implement specific strategies to minimize it. The key point is that waste reduction isn't just about cutting costs. It's about optimizing the entire system for better efficiency and sustainability. This example ties back to our overall theme of continuous improvement and the importance of data-driven decisions. When we apply these concepts consistently, we can achieve substantial resource savings and enhance our operational performance. In conclusion, adopting a risk-based and statistical approach to sampling truly unlocks several key benefits. It helps us reduce product wastage, meaning less material goes to waste, and we become more efficient overall. It also enhances manufacturing efficiency by focusing our efforts where they matter most. Better contamination control is another critical outcome, ensuring the safety and quality of our products. Most importantly, it enables us to stay fully compliant with global sterile GMP regulations, which is essential in our industry. Now, to make all this work effectively, there are some key success factors to keep in mind. First, robust aseptic process validation is fundamental. It confirms our processes are consistently under control. Second, implementing data-driven sampling plans allows us to make informed decisions based on real evidence rather than assumptions. Third, continuous process monitoring helps us catch any deviations early and maintain high standards over time. And finally, maintaining a strong pharmaceutical quality system ensures we have the right structures and culture in place to support these efforts. When we connect these points to the overall theme, it's clear that integrating these strategies not only improves product quality and compliance, but also enhances our operational resilience and efficiency in the long run. For further support, please feel free to reach out to us. We're here to help with any questions or additional information you might need. Don't hesitate to contact us through the provided channels. And with that, I want to sincerely thank you for your time and attention today. Your engagement is truly appreciated, and I hope you found this presentation valuable. Remember, we're always here to support you in implementing and understanding these concepts. Thanks again, and have a great day.